Vectors. Yesterday we had a quick introduction about biotechnology. So today we are going to start learning about vectors. In biotechnology, vectors serve as essential tools for transferring genetic material between organisms, facilitating gene cloning, expression, and manipulation. Here is a comprehensive exploration. So, in biotechnology, a vector refers to a DNA molecule used as a vehicle to transfer genetic material such as genes, DNA fragments, or RNA molecules into a host organism such as bacteria, yeast, plants, animals, or various applications including gene cloning, expression, and editing for these purposes. Okay. So types of vectors can be classified into various types based on their origin, size, function, and applications such as plasmids, bacteriophages or phages in short. Then cosmids are there, then bacterial artificial chromosomes or BACs, then yeast artificial chromosome or YAC, then viral vectors are there, and transposomes can act as vectors, and each offering specific advantages, limitations, and applications in biotechnological research and industrial applications. Next, let's learn about features and components of vectors. Origin of replication or ORI site. ORI site. Vectors contain an origin of replication ORI site that facilitates DNA replication within host cells, ensuring the propagation and maintenance of the vector and its inserted genetic material during cell division. Then selectable markers. Vectors often incorporate selectable markers such as antibiotic resistance genes or markers auxotrophic markers enabling the selection and identification of host cells containing the vector and its inserted DNA through antibiotic selection, nutritional complementation, and phenotypic screening. So, selectable marker is there. Vectors often incorporate selectable markers such as antibiotic resistance genes or auxotrophic markers enabling the selection and identification of host cells containing the vector and its inserted DNA through antibiotic selection, nutritional supplementation, or phenotypic screening. Then, multiple cloning site. Vectors feature a multiple cloning site or polylinker region. Selectable marker is there, then multiple cloning site or polylinker region comprising multiple unique restriction enzyme recognition sites, facilitating the insertion of DNA fragments, genes, or genetic elements into the vector using restriction enzyme digestion and DNA ligation techniques. Promoters, enhancers, and regulatory elements. Vectors may contain promoters, enhancers, and other regulatory elements necessary for controlling gene expression. Multiple cloning sites are there, then promoter, enhancer, and regulatory elements are also there. So these regions contain promoters, enhancers, and other regulatory elements necessary for controlling gene expression, transcriptional regulation, and protein production within host cells, enabling precise control and optimization of recombinant protein expression levels, timing, and conditions. The next about applications of vectors in biotechnology, gene cloning and expression. Vectors serve as essential tools for gene cloning, enabling the isolation, insertion, and propagation of specific DNA sequences, genes, or genetic elements within host cells for studying gene function, structure, and regulation and expression. Then, recombinant protein production. Vectors facilitate the expression of recombinant proteins, enzymes, and biomolecules in heterologous host organisms such as bacteria, yeast, plants, and animals for industrial applications. Therapeutic protein production, biopharmaceutical manufacturing, and research purposes. Gene editing and genetic engineering. Vectors, particularly viral vectors, are transposon based vectors and are used for genome editing, genetic engineering, and gene therapy applications such as CRISPR Cas9 mediated genome editing, gene knockout, knockdown, knock in, and modification of target genes, genomes, and cellular pathways within living organisms and cells. Vector design and optimization strategies. Vector construction and assembly. Biotechnologists design and construct vectors using molecular cloning techniques, DNA synthesis, polymerase chain reaction, or PCR restriction enzyme digestion, DNA ligation, and recombinant DNA technology methods to create customized vectors tailored for specific applications. host organisms and experimental requirements, and vector stability and maintenance. To ensure vector stability, maintenance, and propagation within host cells, researchers optimize vector design, select appropriate replication origins, selectable markers, and regulatory elements, and employ quality control measures such as sequence verification, vector characterization, and functional validation using analytical methods, genetic analysis, and phenotypic assays. Then vector delivery and transformation. Vectors are delivered into host cells using information passing, like transformation techniques are used, such as bacterial transformation, yeast transformation, plant transformation, animal transfection, and viral transduction methods, ensuring efficient uptake, integration, and expression of the vector encoded genetic material within recipient cells for achieving desired outcomes, experimental results, and biotechnological objectives. So, vector size and capacity act as a challenge or limitation, or some important future perspectives are included in that, like vector size and capacity. The size and capacity of vectors impose limitations on the size of DNA fragments, genes, or genetic elements that can be cloned, inserted, and propagated within host cells, necessitating the selection, design, and optimization of vectors tailored for specific applications, target genes, and experimental systems. Host compatibility and efficiency. Vectors must be compatible with host organisms, cellular environments, and biological systems to ensure efficient gene cloning, expression, and manipulation. So, requiring researchers to optimize vector host interactions, cellular pathways, and experimental conditions for achieving desired results and outcomes. Innovation and advancements. Biotechnologists continue to innovate and develop novel vectors, vector systems, and vector based technologies such as next generation vectors, synthetic vectors, self replicating vectors, and advanced delivery system to overcome existing challenges, limitations, and barriers. So, vectors are fundamental components in biotechnology, serving as essential tools for gene cloning, expression, and manipulation, facilitating advancements in research, development, and application across healthcare, agriculture, industry, and environmental sectors. By designing, optimizing, and utilizing vectors effectively, biotechnologists can overcome challenges, achieve objectives, and realize the full potential and benefits of biotechnology innovations, technologies, and solutions for addressing global challenges. So, cloning vectors are indispensable tools in molecular biology and biotechnology, enabling the replication, propagation, and manipulation of specific DNA sequences within host organisms. Cloning vectors are specialized DNA molecules typically derived from plasmids, bacteriophages, or other genetic elements designed to facilitate the insertion, replication, and expression of foreign DNA fragments or genes within host cells, such as bacteria, yeast, plants, or animals, or various biotechnological applications, including gene cloning, expression analysis, and functional studies. Let's learn characters. Cloning vector possesses essential features and components such as an origin of replication, selectable markers, multiple cloning sites (MCS), promoters and regulatory elements enabling the efficient propagation, selection, identification, and expression of recombinant DNA molecules within host organisms for achieving specific research, development, and production objectives. Then, types and categories of cloning vectors. Plasmid vectors. Plasmid vectors are circular DNA molecules derived from bacterial plasmids, commonly used for cloning, gene expression, protein production, and functional studies in bacteria, yeast, and other microbial hosts due to their stability, versatility, and ease of manipulation. And phage vectors are there. Bacteriophage or phage vectors are derived from bacteriophages such as lambda phage, T4 phage, or M13 phage. utilized for cloning large DNA fragments, constructing genomic libraries, and generating DNA sequencing templates in bacteria. Then cosmid vectors. Cosmid vectors combine features of plasmids and phages, facilitating the cloning and manipulation of larger DNA fragments, gene clusters, or genomic regions in bacteria and other host organisms. Then BAC and YAC vectors, bacterial artificial chromosomes and yeast artificial chromosomes, are advanced cloning vectors designed to accommodate and propagate large DNA fragments, genomic libraries, or entire genomes in bacteria and yeast, respectively, enabling genomic studies, sequencing projects, and functional genomic research. Vector design principles. Biotechnologists design cloning vectors using molecular cloning techniques, synthetic biology approaches, and recombinant DNA technology methods to create customized vectors tailored for specific applications, host organisms, DNA fragments, genes, or genetic elements, optimizing vector features, components, and functionalities for achieving desired outcomes and objectives. Vector construction strategies are different. So, vector construction involves the assembly, modification, and optimization of vector components such as OD, selectable markers, MCS, promoters, and regulatory elements using molecular biology tools, DNA synthesis, polymerase chain reaction, PCR restriction, enzyme digestion, DNA ligation, and transformation techniques to create functional vectors compatible with target host systems and experimental requirements. Applications and uses of cloning vectors. Gene cloning and expression. Cloning vectors facilitate gene cloning, enabling the isolation, insertion, and propagation of specific DNA sequences, genes, or genetic elements within host cells for studying gene function, structure, regulation, expression, and therapeutic applications, such as recombinant protein production, enzyme engineering, and metabolic pathway optimization. Molecular biology techniques. Cloning vectors serve as essential tools for performing molecular biology techniques including DNA sequencing, PCR amplification, site-directed mutagenesis, DNA library construction, functional genomic studies, and synthetic biology applications such as genome editing, gene synthesis, and genetic circuit design. Then biotechnological research and development. Cloning vectors support biotechnological research, development, and innovation by facilitating genetic engineering, synthetic biology, systems biology, and genomic editing projects, advancing scientific knowledge, technological capacities. And challenges include vector efficiency and optimization, innovation, and achievement. The efficiency, compatibility, and performance of cloning vectors